Welcome and study with Tanzania Institute of Education.
The aim of this experiment is to separate the colors which are present in natural materials requirements. In this experiment, you shall require 3 beakers, as you can see them here, 2 petri dishes for covering the volatile liquids we are going to use in this experiment, and 2 petri dishes, a pestle and mortar for grinding the mixture, a small beaker for holding the extract leaves which have different colors, filter paper on which to do the chromatographic separation, a teat pipette. The first step is to put sand into the mortar, which will help the process of grinding. If you don't put sand, then this won't grind well. Introduce into the mortar the colored plant material for grinding, and then start the process of grinding this plant material. When you have grinded your leaves to become very fine, like this one, now add ethanol to extract the colored material from the leaves into the liquid mixture. Using a teat pipette, siphon some of the juice into the small beaker and get enough extract like this one. Here we have got our plant extract, which has several colored substances in it. Mount a clean filter paper on a petri dish and produce at the center of the filter paper drop by drop of the extract. In most cases, you must repeat the same spot. You should not put this extract in more than one spot, and when the filter paper is wet, you stop and wait for drying. This extract is a mixture of the colored plant material with ethanol. We have introduced it on the filter paper. The colored rings have started to form. Wait until it dries up before you introduce the spreading material. Now I am using this diethyl ether, in other words called ethoxyethane, which is a better solvent than ethanol, and it will spread further the colored substances. The procedure is the same, that I will have to add the drops at the center of the filter paper. The rings are spreading further and further, moving from the center of the filter paper. Now you can identify different colors or different colored rings from the center outwards. It is expected you will have more than 3 colors. Chromatography is a very important method of separating mixtures, and it is extensively used in research in chemistry, biology, and all the other sciences.